Welcome to our microlearning channel. In today's video, we will be sharing on the importance of auxin transport. This short video frames the central challenge plants face. They must explore complex environments while their bodies are fixed in place, and the hormone auxin sits at the heart of that problem. The images show how uniform auxin produces straight, unresponsive growth, while asymmetric auxin distribution, generated by directional pumping, lets roots bend toward moisture or away from salt, enabling real-time adaptation. Here we meet PIN2, the protein that acts like a directional conduit for auxin in the root, and the illustrations help make that concrete. PIN2 is a specialized efflux carrier concentrated at specific membranes in the lateral root cap, epidermis, and cortex, and its precise positioning drives a basipetal U-turn flow of auxin from the tip back into the elongation zone, sculpting root architecture. The mechanism diagrams walk us through how gravitropism unfolds. Statoliths in the root tip sense tilt. PIN2 becomes enriched on the lower side and pumps auxin asymmetrically, and that shifted auxin gradient slows growth on the lower flank, while the upper side elongates more, producing a bend that reorients the root toward gravity in a coordinated cellular response. At the subcellular level, this slide shows the vesicular trafficking engine that powers PIN2 dynamics, with endocytosis into Brefeldin A-sensitive endosomes and rapid recycling back to the plasma membrane. PIN2 is constantly cycled and can also be routed to the lytic vacuolae for degradation, a balance that functions like an accelerator and brake to tune auxin flow and grant roots fast responsiveness to changing conditions. Post-translational control fine-tunes that efflux machinery, as the diagram of PIN2 phosphorylation and ubiquitination explains. Kinases like pinoid and D6PK phosphorylate PIN2 to establish its polar localization and switch on transport capacity, while E3 ligases tag PIN2 for vacuolar degradation when downregulation is needed. And all of this sits within a network of hormonal crosstalk that shapes root behavior. The final slide ties mechanism to ecological outcome by comparing wild-type and PIN2 mutant roots in a toxic salt patch, and the schematic makes the difference obvious. Active PIN2 localization shifts the auxin maximum to bend roots away from danger, while static PIN2 distribution in mutants fails to shift the gradient, causing blind growth into hazardous zones and demonstrating PIN2's role as the physical executor of adaptive root steering.